Namaskar. Today we will discuss proteins in B.sc Part 3rd Honors. Protein is a very complex natural compound which is formed by combination of many different amino acids. Name of protein is taken from a Greek language word protein, which means primary, that is protein is biologically basis of each living cell. In comparison to vegetables, quantity of protein is more in animals. High proteins are found in mammals, in human beings, hair, skin and hemoglobin, muscle, antibody, enzymes. Some humans like insulin, etc. are made up of protein. It is important to note that except twins, tissue proteins of any two persons are non-identical. General characteristics of proteins. Maximum proteins are high molecular weight containing polymers. Proteins are colorless and non-crystalline. Its melting point and boiling point are not definite. Proteins are mostly insoluble in water, alcohol, and ether. Some proteins are soluble in dilute solution acid and some are soluble in dilute solution of base. Proteins from colloidal solution is water. These colloidal molecules are passed through filter paper but not passed through parchment paper and semi-permeable membrane. In protein, one free acidic and one basic group are present. Therefore, protein molecules are amphoteric in nature. Isoelectric point of proteins are definite. On this isoelectric point, proteins show very low solubility. Proteins are prepared from amino acids, therefore, these are levitary in nature. Oxidation. Proteins get oxidized on burning or not on decomposition or on melting. On oxidation, amine, nitrogen, carbon dioxide and water, etc. are formed. A smell which spreads because of burning of a dead body or decomposition of tiny body animals is the smell of amine. At pH less than isoelectric point, protein behaves like a cation, and at a pH higher than isoelectric point, it behaves like an anion. Denaturation of protein. In the cell or in tissue of any living being, conformation of protein found in specific chemical and physical states are called as simpler or native state of protein. In this state, protein is active in biological form. Chemical bond responsible for the production of secondary, tertiary and quaternary structure of protein are comparatively weak, which easily breaks by different changes in the environment, as a result of which biological activity decreases. This process is called as denaturation. Causes of denaturation. Biological activity of most of the proteins destroys when it comes in contact with the strong mineral acids and bases. Except this, other important reason for denaturation are change in pH 353 to 373 K, temperature, contact with heavy metal and organic solvent, etc. On heating, for example, yellow part of egg on heating coagulates and converts into white denatured protein. Proteins of bacteria also coagulates on heating, and due to this reason only in hospitals instruments used in operation or for injection are first boiled in water, as a result of which all the bacteria sticking to these precipitates from radiations. Protein also coagulates by radiations. This process is used for treatment of cancer by coagulation of protein by organic solvents. Most of the protein coagulates by alcohol and acetone. Due to this reason, while dressing any wood, boiled spirit is used by mineral acids. Inorganic mineral acids like HCl, H2SO4 and HNO3 also precipitates protein. Presence of albumin in urine can be determined by concentrated HNO3. In a test tube, when concentrated HNO3 is added to urine, then in the form of white ring albumin precipitates. This is Halley's ring test of albumin by heavy metals. Heavy metal like HgCl2, AgNO3, etc. precipitates protein. Due to this reason, these metals or salts act as poison for the body because they precipitate the protein which can cause death of human beings. Significance of denaturation. This property of protein is used in clinical curing of disease. Sample of blood or serum taken for analysis of glucose, uric acid and small molecules of medicine is first treated with some special acids. Due to this, maximum proteins get precipitated. These are removed by centrifugal method. After this, supernatant protein-less liquid is chemically analyzed. Reciprocal process of denaturation is called as renaturation. For example, denaturation of protein takes place on heating, but it is cooled down slowly. That is, on annealing it again undergoes renaturation. Classification of protein. On the basis of structure, protein is divided into three categories: simple proteins, conjugated proteins, derived proteins. Simple proteins. Simple proteins are made up of only alpha amino acids. These on hydrolysis give only alpha amino acids. Some of it contains carbohydrate and phosphate radicals. On the basis of its properties and solubilities, these are again classified as follows. First is fibrous protein or scleroproteins. These are insoluble amino proteins and are unaffected by proteolytic enzymes. Fibrous proteins are inoculated molecules which are made up of many coiled peptide series. In this group, silk, wool, skin, hair, nails, foot, connective tissues, and proteins of bone are included. These are divided as follows. Collagens, these are mainly found in connective tissue which are derived from mesenchyme. These are insoluble in water and are unaffected by human digestive enzymes. On boiling with water, dilute acids and bases, these convert into easily digestible soluble gelatins. Pepsins and tryptophan reacts with it. Keratins. These are proteins of hair, wool, foot and nails, etc., which are formed from ectoderm. Myosin. This is a fibrous protein found in muscles. Osin. This protein is found in bone and teeth. Globular proteins. In this peptide series are folded or coiled very easily, very densely. Therefore, their shape is oval or round. In general, these are soluble in water in aqueous solution of salt, acid, base, and alcohol. In this group, enzyme oxygen carrier proteins and ACTH, oxytocin and insulin, etc. proteins and hormones are present. On the basis of solubility, these are divided into following categories: albumin and globin. So this is all about classification of proteins. Thank you.